Okay, so let's look at Mendel's laws. So first of all, who is Mendel? He's this old-looking guy over here. Mendel, Gregor Mendel. So basically, Mendel studied different aspects of a plant. So he looked at the seed shape, the flower color, the seed coating, the tint of the seed coating, the pod shape, the unripe pod color, the flower location, as well as the plant height. So he looked at these 7 traits of a plant and he began to understand these things and then find relationships between how tall the parent plant was compared to the offspring, and he became the father of modern genetics. So Mendel is basically the father of genetics. This is an important, but they sometimes ask this in exams, who's the father of genetics? Your answer is always going to be Mendel. So Mendel came up with 3 different genetic laws. His first law was the law of dominance, and we did go over this in our example over here, our law of dominance. So when a dominant allele is present, it masks the effect of a recessive allele, as we can see, where the little t is masked by the capital T and the phenotype ends up being tall, even though a short allele was present. So basically, that is Mendel's law of dominance. Quite a simple law, and I think we understand it by now. His second law was the law of segregation. This basically states that alleles separate and are randomly divided between different gametes. As we already know, our gametes are our sperm cells and egg cells. So what the law of segregation tells us is that during meiosis, the separation is random and it divides between your egg cells or your sperm cells. So basically, one allele does not depend on another. It separates randomly so that each gamete is different from the other, and this gives us many possibilities of having the same allele in different generations. So it gives us variety as well as the presence of those alleles in future generations. His third law was the law of independent assortment. This basically is that the different characteristics are passed down independently. So basically, this tells us that the gene for tallness is independent of your hair color. So these genes can separate and be passed on independently. So we look at them individually. We look at each characteristic individually according to Mendel's law of independent assortment, because that basically means your hair color and your tallness, it's not linked, it's independent. It's separated independently and it can represent itself independently, which means that you can be tall and still have black hair, or you can be short and you can still have black hair. One does not influence the other. Okay, these are important things that we need to remember when looking at the monohybrid cross. The monohybrid cross is very important. It forms a large part of our exam. So let's look at it. A monohybrid cross is a selective breeding cross that involves a single allele of genes that codes for 2 different alleles of the same body form or part. What this basically means is the monohybrid cross is selective breeding, but it takes two different individuals of that population and it brings them together for fertilization, and then we're going to look at the possible offspring types. So we did look at something similar in our examples here, where we looked at the different genotypes and the resulting phenotypes. So what we're going to do in this one is give it more structure. Okay, so As you can see, this monohybrid cross diagram is done for flowers. So we have a lilac flower and a white flower, and these are the parents flowers. So the phenotype is lilac and it's crossed with a white flower. The genotype of the lilac flower is capital P, and it's crossed with little p, which is a white flower. Then what happens is meiosis. When meiosis happens, capital P and capital P separate to give you capital P, comma, capital P, and these are your gametes. This is very important. This is not PP. This is capital P, comma, capital P. And that's crossed with your gametes of your white flower, which is little p, comma, little p. The reason why these are just single alphabets and not a double alphabet like the top here is because meiosis, which is a process, took place. It's very important to highlight this in your diagram. You never forget your process of meiosis. You write it and then you separate your alleles into single alphabets here. After meiosis takes place, fertilization takes place, and then we draw up our table to represent our resultants. So the gametes from the lilac flower versus the gametes from the white flower. This is your lilac flower, this is your white flower. So your capital P and your little p. Capital P from your lilac flower, little p from your white flower. So there's 4 different possibilities here, and as you can see, they all look basically the same. Our F1 generation, which is our offsprings, have a genotype possibility. Capital P, little p, capital P, little p, capital P, little p, capital P, little p. So what this tells us is basically they're all the same, capital P, little p, because we were looking at a homozygous dominant crossed with a homozygous recessive here. Remember what homozygous means? Both having 2 identical alleles. This is the dominant allele, this is the recessive allele. So whenever a homozygous dominant is crossed with a homozygous recessive, the offspring type will always look something like this. It'll have one dominant allele and one recessive allele. So our ratio in the end is 100% heterozygous lilac, which is capital P, little p. And our phenotype, all are lilac flowers. So this holds true whenever a homozygous dominant is crossed with a homozygous recessive. So let's look at some examples of our own. Okay, so we're going to look at our example for tallness. We've already said the capital T meant tall and little t meant short, but you should write a small key in the corner. So we always start with our P1 generation, which is our parent, and we always start with the phenotype. Don't worry about this. This will be given. It'll tell you Mary and Jake are both tall and this is their genotype. So what you're going to do is you basically write in your phenotype first. So that's tall multiplied by tall. And then you fill in your genotype. These 2 bits of information will be given to you up until here. So after you write that, you're going to put in meiosis, which is a type of cellular division, which we already know. And from meiosis, it results in our G1, our gametes forming. So this capital T, little t splits into its alleles. So you get capital T and little t crossed with, again, capital T and little t. So once this happens, fertilization is taking place and the alleles are going to join, one from each parent. So we follow our lines. You can do it either like this or as a table like this. The table can become more confusing, and it's also time consuming. So I would highly recommend doing it this way. So you take each allele and you cross it with each of the other alleles from the other parent. So follow our blue line, capital T from parent 1 and capital T from parent 2. So that gives us our genotype of capital T, capital T. Following our black line, capital T from parent 1 with little t from parent 2. That results in capital T, little t being our genotype. So we've already done allele 1 of our parent 1 with both alleles from our parent 2. So we move to allele 2 of our parent 1 and we follow our red line. We're taking little t and capital T from here. So we end up having capital T, little t as our other genotype. And then we follow our last line, which is our yellow line, and we get little t from parent 1 and little t from parent 2, which becomes our final genotype of the offspring, which is little t, little t. There cannot be anything more because there's only 2 alleles from parent 1 and 2 alleles from parent 2. So there's only 4 possible outcomes in this situation. And then once we know our genotypes, we can then write out our phenotypes. We know when there's a capital T present, we automatically know it's tall. So we're just going to write. So that's tall, that's tall, that's tall. Here there's no capital T present, it's just little t, so it's short. And then we can get our ratio. So there's 3 tall is to 1 short. Therefore, there's a 75% chance that the offspring from these 2 parents will be tall. Okay, so I would suggest you go over that one more time to make sure you understand what went on specifically around there. And we can do another example for having a cleft in your chin. So let's look at what a cleft looks like. So this is a picture of someone who has a chin cleft here on the left, and this is a picture without a chin cleft. It's a smooth chin. So in this situation, our dominant trait is having a chin cleft. So we're going to do our diagram for chin clefts. Okay, so like previously, we start with our P1 phenotype, which is cleft and no cleft. Like I said, you will be given the phenotypes as well as the genotype of the organism. So we proceed to writing out our genotypes, which in this case was heterozygous crossed with homozygous recessive. Then meiosis took place and we resulted with having gametes of alleles capital C, little c, crossed with little c, little c. So from there, fertilization took place, and once again we follow our blue lines, our black and red and yellow lines to get capital C, capital C, capital C, little c, little c, little c, and little c, little c. And our phenotype was 2 having clefts and 2 having no clefts. As always, our genotype will always have 4 different genotypes. And our ratio is 1 cleft is to 1 no cleft. Therefore, there's a 50% chance of the offspring having a chin cleft. Okay, so now that we've got 2 examples down, I want you to go through some examples of your own. So here we have our exercise. So you're going to do a monohybrid cross and find the ratio of the following. Okay, and there's an image of dimples and no dimples, and note that dimples is the dominant trait. Okay, so that's our first question where we do 3 different monohybrid crosses. Then there's question 2 where we look at free earlobes and attached earlobes, and you're going to do a possibility of that. And question 3, which is very interesting, I want everyone to attempt it, and then in our next lesson we're going to go over the trend. There is a trend. I would have discussed it with you guys now, but then I want you guys to see it on your own when doing the monohybrid crosses on your own. All the best, and hope you get it right.